As demand for advanced therapies continues to surge, the biopharma industry is under pressure to innovate faster in high risk areas like drug discovery and cell therapy. And these fields face persistent challenges such as high costs, long development timelines, and complex regulatory landscapes. But to that end, MaxCyte has introduced a new high throughput electroporation platform that streamlines genetic material delivery with speed, precision, and scalability. Hello, everyone. I'm Miles Skoda, and this is the CellAndGene.com virtual event series. And today we're talking advancing therapeutic discovery with reproducible high-throughput electroporation. So joining me for this presentation is an industry expert from MaxCyte, head of R&D and product innovation, Dr. Grace Dharamaja, who is going to explore how the new software enables fine-tuned energy delivery across diverse cell types, including hard-to-transfect immune cells, and walk through real-world applications such as optimizing non-viral CAR-T engineering using CRISPR-Cas9, demonstrating high-throughput efficiency with consistent reproducibility. We are going to be holding a Q&A session as well following this presentation, so please feel free to submit questions as we go along. We'll get to as many as possible during that Q&A, but any that we don't get to, I'll pass along to the team here for potential follow-up after the event. And then just a bit of housekeeping. If you run into any issues today, please try refreshing your browser first. If your problem persists, you can submit a question through that Q&A, and I'll help get you back on track. And then finally, this event will be available on demand, usually within a day, often much less. You'll get an email with a link to it, or you can just use the same link that you used today. And so with all that said, let me turn it over to Grace to get started. Grace?
Thank you so much, Miles, and thank you for hosting us. We really appreciate it, and obviously enjoy a lot of the content that you all provide to the industry. So today, as Miles mentioned, I will be speaking about a new instrument that we have launched here at MaxCyte, which should address a lot of the early stage discovery work that scientists do in the cell and gene therapy space, but also beyond that as well. So this is the your typical sort of disclosure slide. Thank you for bearing with us through that one. I'll begin with sharing a little bit about the history of our company and how we got here and what industry or what markets we typically tap into at this time, for those who don't know us. For those who are active customers of ours and we speak to regularly, thank you for working with us, and we appreciate a lot of the challenges you're overcoming to move this industry forward, and we're excited to see what you will produce in the coming years. So MaxCyte has been in the cell and gene therapy space for the past 26 years. We started in the beginning, we focused on enabling a safe and efficient cell electroporation platform technology. This platform is known as the ExPERT electroporation technology, and it's known for being application agnostic. So you are able to scale the production of your therapy, whatever that might be, depending on what your cell type specifics are, as well as your payload or your cargo, all the way from concept to the clinic on a single platform. And that's sort of depicted in this image here, where we are really just pushing this field forward with our technology in the workflow. As you can see very clearly on your left, you have the sort of workflow where you're collecting from the patient, engineering the cell, and putting it back in the patient in order to treat a disease. And then on the right, you're kind of seeing that same ex vivo platform a little bit more in detail, where the cells that we take out of the patient that are those primary cells, you can modify them with the payload, as we mentioned, of your interest, go through our system and really get those cells ready to go back into the patient. So that's really the exciting part of what we're able to support as we go through, we go through the years of discovery at our company. Now, the more sort of details of where we actually fall in the specifics of our instrument is, you might be aware of the last three instruments that you're kind of seeing here. So the ATX, the STX, and GTX. These instruments have really been designed for addressing different needs in either cell therapy, drug discovery, antibody production, or vaccines. The newest instrument, which is the one that you see on the far right, is the DTX. And it's really aimed to target folks doing the early discovery space. And the key part of that is the fact that it allows electroporation of 96 wells, of 96 samples in under 3 minutes. So that's a really exciting launch for us, and an exciting enablement that we're providing to the community. And we've already gotten a lot of excitement. Folks are trying it out, and I'm going to take you through a little bit of how we're shaping this industry. We have a couple of these poll questions inserted in here. So this is one of those, and take your time with answering it. It'll remain on your screen. I'm going to keep going on with my talk, but you'll just see a few more of these come through. So in this case, as you can imagine, we're super excited and understanding what applications folks are currently working on. We'd love to, one, develop workflows that make your life easier when you do try out our technology. But what's more, it's also telling us kind of where the industry is moving, and that's really exciting for us to know as well as a general scientific community. All right, so just to give you a little bit more confidence for those who might not know about our platform and its participation, if you will, in kind of enabling this next generation of cell therapies. I want to take you through a slide that we do tend to share pretty commonly out there. So some of you may already know this, that we do have an FDA master file, as well as other regional equivalents that support our technology in different parts of the world. And so that's extremely key, because now you can simply citing towards submitting all your regulatory filings. And what you can actually see from this diagram is that the ExPERT has in fact enabled several clinical trials. And in fact, we've even been used commercially with a drug therapy called Casgevy that has been developed for sickle cell disease. And so that is something we're very excited about, and we're super grateful for our customers who work with us and participate in all this. They're doing all the hard work. We're hopefully enabling that by providing a scalable platform that is really ready to go into those regulatory filings with you. So just a few highlights here to show that even in the commercial, or in the clinical space, I should say rather, that we have been supporting a wide variety of not just cell types, but also the types of modifications that are happening to those cells and the types of indication they're servicing, basically. So you can see the types of indication below, which is sort of these genetic diseases. You can see hematological malignancies or hematological cancers, as well as solid tumors and autoimmune diseases. And then these are typically captured by your HSCs and T cells that you're seeing here, mostly in the circular diagram. But we also have a few therapies that are in other cell types, which is pretty interesting as well. So this is just sort of to give that confidence that we've been in this space for quite a while, and we're continuing to help folks progress through to the clinic with what they're developing. Now, it comes on the back end of the fact that we've been around for 26 years, as I mentioned when we first started. And really that the development of not just the instrumentation, but the actual electroporation protocols, which is really kind of the foundation of what we've built into these instruments, they've been in development for these 2+ decades, right? And so really highlighting that as the customers migrate and go through the clinic and develop their therapies, we've been along that journey with them, given how long we've actually been in the industry. Some of the key things I want to highlight here is, of course, our ExPERT platform came into being in 2019. To some extent, it's a bit of a branding. However, a lot of the electroporation protocols that have been developed are the same from those early days of the company. And we continue to develop those protocols depending on what other cell types that might be unique that you want supported, or if you want to see a different biological outcome when you treat those same cells. So we're able to continue to expand with the customer. And what's really cool here is, as we kind of march through those instruments that are used in the clinic or in process development, we clearly saw a need that we have these customers who would love to be working at this larger scale for screening and really looking at the various components that go into a cell therapy and being able to screen the various categories individually, right? And they don't need the level of sort of high level production that we had going in ExPERT. What they needed was the ability to be able to do many, many more samples and do it in a cost-effective manner. And so that's what's really given rise to the development of the ExPERT DTX, as we're calling it. So that is another member of our family, new member of our family, and we did launch that earlier this year, and we are in very active in demos. We've even sold a few instruments, and we're continuing that process, and we can see the adoption happening with it. So I'm going to take you through a few key highlights of our data that we've generated with that instrument, but also I want to show you what that instrument looks like in a little bit more under a microscope, if you will. So here what you're seeing is just a closer up look of the instrument, fairly expected components, especially if you've worked with electroporators before. I'll start with what we call our consumable or the process assembly, which is the R50 by 96. So this is a high throughput process assembly that allows you to run 96 samples in the 3 minutes. Again, as I mentioned, I will spend a little bit more time on showing you the details of this in a second, but it sits on top of, so the consumable sits on top of what we call our processing assembly module, or our PAM. And that's the smaller black box that's sitting on top of what will eventually be the brains of the operation. Now, what that smaller black box, or the PAM, does is it allows us to hold the processing assembly and deliver the energy that we need to do in it. Deliver the energy that we need to deliver into the samples. What it is, it's connected on the back with wires that allows it to speak to the brain of the instrument or the platform, and it's acting as a holder. And it looks very familiar to PCR machines, things that you would see in the lab. And that's really the intent of this design, is that it should be a very intuitive way that you interact with the machine, as you do with other instruments and machinery that we already have in our labs. And so if you move to the electroporation module, which is what you're seeing at the bottom, the guts of this instrument is exactly what we have in all the way up to our GMP instrument that goes to the FDA goes with the FDA master file. So meaning that we have not compromised on quality here when we built this instrument. We want to be able to deliver energy in a precise and controllable way. So we ensure that we can do that with the base module. And in this module is also where you can control the operation of the instrument. So like our other ExPERT instruments, it's a very easy to control software or a user interface, which is what you're seeing pointed here, where you have the option of, setting up an experiment, pulling up a previously set up experiment. You can go in and edit. And I'll show you what a screen looks like in subsequent slides as well. Now, as promised, actually, I believe we have a poll question again. So I'll let that poll question cover through. And so, obviously, we've just talked about a cell engineering process. It's really would love to hear y'all's thoughts on what is the most important part of cell engineering that you believe needs improvement still, right? Obviously, it's a very loaded question, which will be great to hear what people have to say. We're hoping we've addressed some, if not all of these, with what we've developed for the research discovery group. So quick highlight of what we spoke about in terms of the instrument. It allows you to sort of do that sort of high throughput screening that we mentioned, particularly because that plate itself can be pipettes as well as automation. And so it allows you to sort of scale in that way and screen quickly in that way. But also things that we've kept in mind as we, just passed that poll question is we wanted to make sure it was cost effective so that you could afford to screen 96 wells at a given time. But we did not want to sacrifice the quality that MaxCyte is seen as with all of our other instrumentation. And we wanted to make sure you had access to all of those electroporation parameters that are available on all the way to our GMP instrument. And so that's what we've really enabled with the DTX. Now, My favorite part personally is really looking into that consumable, which we call the process assembly. So we've designed it in such a way that we maximize for quality while giving you the best sort of cost associated with it. And in order to do that, we we created this strip format. So every single strip or column is independent, meaning you don't have to run all 96 wells. You don't have to run all 12 strips. You can run one strip. And so the instrument absolutely works on that configuration as well, where you would choose to run fewer than the 96 wells. Now, things that we thought about here, we wanted to make sure you can actually retrieve your sample back out. And so the curvature of the chamber that your cells and payload will sit in is in a manner in which the pipette tips can retrieve your samples. And in the case of when you march towards automation for that, again, we have a little bit of familiarity, particularly with the types of tips that should be used here. Now, we have validated internally 20 to 50 microliters. The capability of this, the chamber is a little bit higher than the 50 microliters, so it can actually go well up to sort of the 90, 95 microliter space. And folks can test that, on site as they demo. We're continuing to work in these smaller volumes because we know when you're screening so much, we want to make sure we're not using a ton of cells that's going to become extremely costly and prohibitive to being able to screen what you'd like to screen. So that's why we try to optimize in these smaller cell volumes. And so a lot of the work you'll see downstream of these slides will be very much focused on the 50 microliter space. But it does enable you to effectively screen, between hundreds of thousands to 10 million cells. And that's more than enough for doing, we found, speaking of folks, it's sufficient to be able to do some of the downstream analysis that's required when you're in these sort of screening processes or testing processes. So I want to give you a quick look at what I mentioned before, which is of the kind of the active screen when you're programming your experiment. Now, what's really interesting about this is you can actually program this outside of the instrument. So we do have a software that comes available with this instrument where you can actually do this on your computer screen, or just take it over to the instrument. So you're not sort of cowering over the instrument trying to do all of this. So going to what you're seeing on the left-hand side at the top, that's really the programmable portion of your experiment here. So what you're seeing is each of those columns represent the strips that we talked about. So very intuitive. When you're clicked into one of those, you can click a whole strip, or you can go in and actually click a specific well. Now, when you do these activities, you can see on that same panel on the right of it, where you can actually type in what you want labeled there. So when you start hovering over those spots where there's a little pen, a keyboard will pop up and you'll be able to type. Now, of course, if you're doing this on a computer or your laptop, that's very easy to do, right? If you're doing it on the machine, it's a touch screen and the keyboard pops up and you can move it up and down. Now, on the lower part of that panel is something we're very proud of. So some of the thoughts we've heard from customers is, I'd really like to be able to do a little bit of tinkering on my own. We understand that you're developing full-blown protocols for all these unique cell types, but what if I could take one of those and play a little bit left or right of it? So we've enabled that. That's the call out you're seeing on the bottom left-hand side. And so here, if you look really closely, what you'll see is we're trying to color coordinate some of these. So what you see is the colored boxes along the bottom match the graphs. But before we move there, I'd like to take you along what we call our toggler. So it's set up at K562. Meaning it's the electroporation protocol for K562. Now, when you take that protocol, what we've enabled is that there's 3 major units to the right that allow you to increase the energy, the total energy flowing in to the chamber, or you can decrease it. Again, 3 major units to the left. There's finer movements as well, which we've highlighted. Now, if we move down to those colored boxes, the K562 that's green is representing the DTX electroporation protocol and cells, the experiment that we did. Now, the one below it, which is this purple, is the control that we did on the GTX. Now, remember, the GTX is our GMP instrument, which we've had on the market for several years. And so that GTX acts as sort of the foundational place where we've developed a lot of our protocols. So we always go back to that instrument and we compare against it when we're developing new technologies. Now, the other subsequent colors that you're seeing is referring to parts of the protocol that were the electroporation protocols that were chosen that are either higher energy, those lighter blues that you're seeing, or lower energies, which are the darker blues that you're seeing. So now if you go to the graph with me on your right, there's 2 graphs here. So everything is K562. What you're seeing is the cell viability on your left, and you're seeing the transfection efficiency on your right. Now, no surprises here. K562 is an immortalized cell line. So we don't expect to truly hit too many concerns when it comes to viability, even as we push up the energy being delivered into the cell. Now, generally, you can imagine that when you alter the core energy being applied, the higher you go, the less happy the cell probably is, right? But with an immortalized cell, it'll recover because there's so many other cells that can continue to replicate and actually repopulate, right? With primary cells, this is not the case, and we'll share some of that information as well. And so when you go to the transfection efficiency, that's where you see the interesting story, right? So what you'd want to do is center yourself on the purple and the green. So those 2 items, what that's showing you is the GTX, the purple, and the green, the DTX equivalent. So as you can see there, not only are the error bars pretty tight, but they're pretty close to each other generally. So the transfection efficiency that we're achieving is similar between DTX and GTX for K562s, as we would have, and the transfection efficiency readout that we're looking at here is coming from a GFP plasmid. We've effectively used 50 micrograms per milliliter in this process. Now, let's go to the higher energy. What you can see is we're pushing up the transfection efficiency, meaning the percentage of GFP positive cells, in a very stepwise manner. And then what happens when we lower the energy? We're pushing the transfection efficiency down. So, and this holds true, as you can see, as the cells go from 24 hours to 48 hours to 72 hours. Now, this shows you the basics of what you want to see with an electroporator that we're developing. We want to make sure that these basic controls that we've programmed into the new electroporator can work in a predictable manner. And that's what this has shown us, is that we're able to do that. Now, I know a lot of you are probably going to be wondering about the primary cells. Just give me a couple more slides and I'll get to that. I just want to get through a few more sort of technical parts about the platform that should give you a little bit more confidence in it. Now, one other thing that we did is we wanted to make sure that you didn't lose that reproducibility that you get with our instrumentation at the GMP level. So we want to make sure that when you're running this instrument, it gives you the same results over and over again. Now, that's pretty easy to do with immortalized lines and something like K562s. But what we wanted to do is show it in something a little bit more challenging, something like T cells. And that's what you're seeing here. So on this slide, we have 32 replicates, and those are the individual dots that you're seeing on each one of those bar graphs. And then what we show you from left to right is viability, transfection efficiency, and then the MFI, or the GFP expression, as we're referring to it here. And so what you're seeing here is how tight the viability is with T cells, how tight the transfection efficiency is with T cells, as well as how tight that GFP MFI is with T cells. So here we are expressing a plasmid that's expressing GFP under a CMV promoter. It is about 4.5 kb in size, and we are electroporating 100 micrograms per mil. And so that translates to about 1 microgram or 5, because you're seeing the different volumes here, right? So we did 20 microliters as well as 50. And if you remember, that's kind of the bookends that we're sticking to, because we think that's a pretty valuable space for folks doing screening work to be in. So that's the reproducibility part of it, meaning that we have not compromised that from our GMP system. And the last sort of technical development slide that we care about is, we hear this a lot, right? When you're working in a 96 well system, there's often really high chance of cross-contamination. And so what we're showing you here is a sample. We've done this several times. As you can imagine, we have 32 replicates in that previous slide. But what we wanted to really show you here is when we load no cargo controls, which are the white boxes, right next to samples that have cargo at high concentrations, even that 50 that you're seeing microgram per mil, that's a pretty high concentration for us. And so what you can see here is that there is no cross-contamination, because when you go to the GFP expression, you're not picking anything up above sort of the background noise. And the ones that have GFP are clearly showing you GFP positive cells that are well above, 88%, right? That's kind of the lowest number that you're seeing there. So really, this instrument has been developed for you to really use your creativity in what you want to screen. And we've thought about all of those technical challenges that you may face, and we wanted to make sure those were not impacting you. So reproducibility, this type of cross-contamination, some of that automation I spoke about that we're working with customers now to make sure these systems can also play into that. And, being able to do everything from easy cell types to cell types that are from a primary tissue source and much more challenging to handle, and not as malleable to an electroporation system. So that brings me now to the case study, which is probably the most interesting part about this, and allowing us to really dive into some interesting things that we've done with the instrument that should hopefully resonate, because these are things we see our typical customer base as well as folks in the industry working on. So most people kind of in that immunology cell therapy space should know about the TRAC locus, which is your T cell receptor alpha chain. It's a very common target for cell engineering, mostly because it serves really great for a place where you can knock in whatever you want expressed in that T cell, and ideally have that T cell recognize that antigen and be able to, release the T cell activities on it, right? And so some of the advantages are listed. Be as familiar is that it's expressed endogenously under a strong promoter, but because it's an endogenous promoter, it's mimicking that natural TCR expression that you would expect, and therefore it's contributing to a uniform CAR expression, and it has sort of a stable CAR T functionality that we're all aware of. Now, if you apply this to an autologous space, it also reduces your graft versus host disease concerns that come up. Now, folks that we've on this, we're just highlighting a few items, right? What is the type of things that people want to optimize in their cell therapies before they start going down a manufacturing road. And so naturally, the first thing that comes to folks are optimizing your physical components independently. So meaning the nuclease and the guide. And there's so many more things under there, right? We've just sort of touched on the variants that folks have been producing for their nucleases. If you look at some of the deaminases that are coming out, there's hundreds, thousands of them out there now, right? And so being able to screen these is really what we're thinking about. Being able to screen any or many nucleotides one at a time and really look through what's going to work for you, we want to be able to enable that. if we're looking at this knock-in phenotype, which is what we're doing for this track locus case study, I think it's really important to also screen not only what's the source of your HDR template? Is it single-stranded? Is it double-stranded? Is it circularized? Is it linear? All of those aspects, but the actual sequences themselves, the actual like overhangs, all the things that you can actually work on for an HDR template here, we can enable that here in this instrumentation. And generally, people want to also look at concentrations, molar ratios, and all the other things that are maybe not as exciting, but definitely very relevant to sort of being able to scale up. And so that's really what this case study is looking to illustrate, is this whole process of being able to take this very commonly used track locus and screen CARs and the components that go into creating that CAR knock-in. Just a really high-level overview of the workflow itself. I'm sure many of you are not only familiar with this workflow outside of operation, assays that we use. You want to be able to prepare your samples for screening, so get them into the right concentrations, either create the RNP, right? So be able to combine the guide and the nuclease in one tube, all of those things. You want to load those samples in a 96-well plate that you're very familiar with, where something as simple as a 96, as simple as a multi-channel pipetter can work. So you want to be able to do that. Now this is the part that's specific to MaxCyte and the DTX, which is you need to be able to load that now onto the DTX, set up your experiment, which we went over, run your experiment, which takes a couple of minutes, as we mentioned, and then be able to unload your samples to culture, expand, and analyze. And so a very general workflow of what typically scientists are doing in the space. Now, I want to dive into the most exciting side of things, which is the data itself. So we took a stepwise approach to this, where every step we took away a variable and moved on to the next one. So we standardize something after we screen it, and we add that knowledge to the next step, and we continue doing that until, in our minds, we get the perfect workflow to edit this locus. So we start with the guides. It's a natural place to start. Most folks do start in that space. And so here what we're doing is, hopefully what you can see in these slides here is that I'll show you all the study details, as we call it, or the methodology. And then that's on the left, on the right, what you'll see is the actual data output. So here we're using Cas9. So we don't want to be varying both the nuclease and the guide at the same time. We're doing that separately. Now we know Cas9 typically tends to be a really good nuclease to use. So if you're going to find your best guide, likely through Cas9, right? So we went ahead and screened inactivated T cells, 5 different guides for the track locus. And we also screened 3 different RMP concentrations, which are represented by the shapes. So 0.5 micromolar, 1 micromolar to 2 micromolar. And so what happens here now is you can do sort of this XY comparison of what's the best viability to the best track knockout I get, right? So this is a knockout screen because you're just looking for the functionality of your guide. Now, ideally, in this graph, you want to be in the upper right-hand quadrant. So highest viability, highest knockout, right? And so what we're showing here is that guide 5 from a track knockout perspective performs the best no matter what the concentration is, because that's your green points, whether it's a circle, square, or a triangle, right? But what you're seeing is even within the concentrations, there's a bit of difference between the viability of each of these concentrations, right? And so notice that our y-axis is actually a little bit zoomed in. So it's between 76 and 90% is the viability we're showing here. So here, that difference between 78 to sort of 89-ish, right? It's a 10% difference. people may care, right? But we're happy that track 5 is clearly a winner, right? And you can look at the other ones for a little bit more patterns, but what we're noticing here is this is an empirical experiment. This is an empirical process for us. We had these 5 guides, we tried them out, and we know that the guide 5 is the best one to move forward with. Now that we know that, we want to be able to screen the best nuclease to move forward with. And so in this case, we had to get a little bit, we have to understand a little bit more here. So we have a couple different types of nucleases, meaning we have Cas9 and those of the Cas12 variants. And that's what we're seeing with nuclease A and B kind of fall into the same category as Cas12A. So we're screening 4 different nucleases here. We have the same 3 RMP concentrations because, hey, we have a 96-well plate. We might as well be able to screen the concentrations. You just never know, right? But the guide is moving forward as one guide, which is track 5, which we cleared at the top at the previous slide. And so what you're seeing here now is the same thing. Look for the color pattern across the 3 shapes, right? And so here what you see is Cas9. Not a surprise, right? So Cas9 has performed the best in terms of track knockout, and for the most part also does really well in terms of viability with your no EP control hanging out up here. Now, each of these were done in triplicates, just as the other slide, the data from the previous slide was. So we're pretty confident in this data as we sort of go through this iterative process of saying, all right, guide 5, Cas9, what's the next thing I'm going to screen? Well, you still have a pretty important component left here, which is your HDR template. So typically, folks, I mentioned to you, you could screen all the different formats it could come in, but one of the things that's pretty commonly screened is the concentration at which to use that HDR template. And so that's what we did here. And so what you can see is we took track 5, we took Cas9, all the ratios are maintained from the previous experiments. We went this time with 1 concentration because if you recall from the previous slide, that concentration did pretty well. So the 0.5, so the 0.5 concentration is sitting up here, right? So we went with that concentration. And here what you're seeing is what is the impact of the concentration of the HDR template here? Now, and what you're seeing is the highest concentration gets you the highest knock-in with a reasonable amount in terms of viability. Because if you start going too low because you're trying to get to higher viability, you're not going to get much in terms of CAR knock-in. And on top of it, remember, this is a zoomed in y-axis, so you're not gaining that much more in viability. So really just to give you an opportunity to see the potential of what you can do in this instrument. Now, for all of this work, because it was in primary T cells, we used what we call ETC4. So those who work with us know exactly what that protocol does. A lot of folks working in the T cell space use that protocol on our instrumentation. Now, that's very key to know that that same protocol that you're using on the GTX, STX, or ATX can now be used on the DTX. And it's that same electroporation protocol. So you can now confidently move forward with the results you got here, which are highlighted in a later slide, but you can confidently move forward to the GTX. So that's what we're doing here, and that's what we're showing you. Now, we've taken all those conditions that we've optimized on the DTX, and what we've done here is we've gone ahead and taken a look at how does that perform compared to the GTX, which is our GMP instrument. We added 1 additional variable here because we think it's a really important variable, and that's the variability that comes from donor to donor. So we've done 3 donors here. We did all of those in triplicates, and we did it across 2 instrument platforms, the DTX, which is the new instrument we launched today, or that we're sharing with you today, and the GTX, which is the GMP instrument. Now, we've also done it with respect to a no cargo control as our TR right? Those are the 2 things we're looking at here. Now, what happens is something to show you is, regardless of, whether there's EP and if there is EP with cargo, on the GMP instrument, that viability stays the same. Now, with the new instrument, the viability is a little bit lower. That's the sort of, that's the sort of compromise we made to be able to make it cost effectively to some extent and to make it allow you to screen. But what's key here is the patterns that you see on the GTX on the DTX, meaning when you electroporate here, you can move to the GTX and do those experiments and get similar results. And that's really important here. Something to also keep in mind is at times the donors may behave differently on the instrument. And that's just what we see commonly here. We see that the donors can have unique biological properties that are pulled out differently on the GMP instrument that's being built for a clinical purpose and the screening instrument that's being built for a discovery purpose. But what's key here is that between these two instruments, you get more than enough cells to be able to do your downstream assays and get that work done. It also allows you to identify a good donor. So if you're going to do some screening work, you might want to focus on getting to a donor that gets you to a reasonable CAR T cell yield such that you can do a lot of that downstream work. So kind of taking it all back, what we've allowed you to do here is really screen through through a few possible parameters that come to mind for us when you're screening for a cell therapy that you're developing. Now, the key here is that these are parameters that we've thought are necessary and wanted to share a story with, but as we spoke about a few slides ago, there's a whole other range of parameters you can test. And there's so much knowledge that comes from individual labs and groups that they know there's other parameters they want to screen, but they might not be able to do it because they're not able to scale that up on the same sort of instrument platform. We're allowing you to do that. So the optimal CAR T production, develop and optimize and screen through on the DT to the ExPERT GTX, allowing you to make that trip into the clinic, that path to the clinic, much more seamless. And so hopefully we conveyed that with some of these slides that we shared with you today. And I'm going to leave you with one last poll question. And so I'll leave that here for those who are participating in the poll questions and be able to get some answers from you. And I'll hand it back over to Miles.
Grace, thanks so much. This was awesome. A lot of great insights. It's awesome to see the new instruments you guys have and everything it can do. So audience members, we're going to take some questions here. Just a couple minutes to take a few questions. If you do have questions, submit those through that Q&A box on your screen. And then do please take a second to respond to the poll questions. If you'd like to get more information, we'd really appreciate that as well. And so with that, let's get into these questions. So the first one here, does the instrument require any specific 96 well plates, and is it compatible with automation?
Yeah, thanks, Miles. Great question. So from the consideration of does it require custom plates? Yes. So those were the plates that we showed earlier on in the slides. This is a 96 well plate that the strips are removable. It's necessary because there's electrodes on the plates that facilitate the electroporation. But again, as I mentioned, we've really taken into consideration the cost associated with this, and we've ensured that it's reasonable for you to do the work and scale in a 96 well format very easily. And so those consumables actually come from MaxCyte. When you first buy the instrument, it comes with a pack of those, and then you can just buy those packs repeatedly as needed.
Okay, yeah, perfect. Let's see, next one here. So what cell types have you tested on the DTX so far?
Yeah, sounds perfect. So we've done primary T cells, primary HSCs, we've done iPSCs, and then we've done some cell lines, which include K562s, which you saw today, CHO, hex cells, Jurkats. We've also done, externally, customers have done things like primary hepatocytes and that type of cell type as well. And so that list will continue to grow. as you know, we do work internally, as customers do work as well. And we're seeing, you know, we've started with the more challenging ones, and what we're seeing is it's pretty transferable. And so we have confidence that the subsequent cell types we test will also work out, and we'll make sure there's a workflow that works for those.
All right. Yeah. Great. Let's see, maybe one or two more here. So what is the efficiency of the system other than T cells? They said, you know, something like NK or other immune cells. Do you have data on that?
Yeah, so we have some stuff on HSCs. I think maybe that would be the first one. Let's start with iPSCs. So iPSCs, we get about sort of north of 80, 85% transfection efficiency. It's close to 100% when it comes to mRNA. It's the plasmids that tend to be obviously a little bit lower. On the HSC front, we have percentages that range in the same similar sort of numbers you're thinking about. So mRNA is sort of north of the 95%, and then plasmid, I would say, is north of they're in sort of the 60% range. But again, depending on playing with concentrations and things like that, and we have multiple HSC protocols as well. So you can also screen through those protocols to see which ones would work better for your donor and your cells.
Okay, awesome. Yeah, thanks, Grace. Let's see, another one here. So why choose the DTX over other available high throughput electroporation platforms?
Yeah, that's a really great question. You know, I think the value that we bring is those sort of early slides we saw. We have quite a few assets being developed on our instrumentation that are in clinical trials. Not only that, the Casgevy is a perfect example of a actual asset that's commercial that is developed on our instrumentation. So you're basically getting access to not just that knowledge and the development work that we've done in 26 years to get to this point, but on top of that, all of those electroporation protocols that are available to these folks marching towards the clinic, they are available at the early discovery stage with the DTX, and they are the exact same protocols. So showing you some of that work where the DTX data on the DTX, or cells being electroporated on the DTX and cells being electroporated on the GTX looking so similar, that's a really common pattern that we're seeing. A lot of folks that are demoing our instruments right now, they already have our other ExPERT instruments, either the ATX or the GTX, and what you're seeing from them is when they do a head-to-head comparison, they're able to translate that data. So it's very seamless in that way, and I think that's what makes it so unique for folks to be able to use. The other part is that toggle function that we have on the electroporator for the DTX. What's really cool about that is it sits on top of our many, many years of research and expertise that we have for these electroporation protocols, but we've opened it up to you a little bit so you can move to the left and to the right of where you are. That gives you a little bit of optimization window that's in your own hands if you need to personalize that electroporation step in your workflow for your processes. So that means, and that if you decide to toggle up or down, those protocols are also available for you on the GTX. You can transfer that up as well. So it makes it very seamless, right? Just having to use that word again and again. You really can just go from discovery to manufacturing. So I think that's why you should choose this. We've got that A to Z solution now.
Awesome. Yeah, thanks, Grace. Let's see. So another one here. What about? Oh wait. Okay, here's this one. So the independent strip control is a big highlight. Can multiple different electroporation protocols run simultaneously across strips in one run, or is it limited to one protocol per run with different cargoes?
No, so each of those strips can have a different electroporation protocol. That's the beauty of sort of that design, the UI or the user interface that we've created. So now what you can do is test different protocols, whether you're toggling or using, what we have is ETC3, ETC4, which are the T cell protocols. You can actually do those across a single experiment in a single 96 well plate. Now, of course, you can actual components or reagents that go in, whether it be concentration sequences, all of those things, but it really does give you the full matrix of what you can play with. And truly, it's just up to you. Now, we've also done a lot of that heavy lifting and work. So please also come talk to us and, let's work together so that you're not doing an experiment that's like 20,000 samples and we can pare that down to something more reasonable.
Yeah, perfect. Let's see, maybe one or two more here. Thanks so much, everyone, for all these great questions. Certainly submit some more if you have them. Again, if we don't get to yours, I will pass those along to Grace and the team here for some follow-up after the event as well. So let's see. So what about T cell subsets or resting T cells instead of activated? How do you adjust protocols for those?
Yeah, so we actually have developed those again on the previously existing instruments, if you will. And so those are absolutely available on the DTX. A small sort of biology for you all. Generally, resting T cells, smaller, they're not in mitosis, so they actually require more energy. And so that's really the difference between the protocols for T cells and resting T cells. And we've developed few for both of these categories. And so what we can do is easily take those protocols and put them on the DTX. They are already on the DTX, I should rather say. And so you can screen for resting T cells, any sort of work that you have associated with that cell type, you can do that. And in fact, we're doing that in-house as well. So you're able to, we're able to kind of help you on that journey as well, because we have a lot of that workflow. I'll also add to that a little bit, Miles, is some of the things that are required here is when you increase that energy, you do need to think about what your buffer strength is, meaning what's the conductivity that's going through the well. And so that is often manipulated or managed by the buffer's ionic strength. And so there's a lot of buffers out there that folks use. MaxCyte has historically only had one buffer, but as the market has moved towards the need for these higher energy protocols, such as in the resting T cells, what we've also now done is developed a low conductivity buffer or a buffer with lower ionic strength, and you can apply that to all of your workflows. And that applies for all the instruments, not just the GTX, not just the ATX to GTX, but also the DTX.
Let's see. So does the DTX integrate with common automated liquid handling systems?
Yeah, that's a great question. Thank you for that. And so I did allude to that a little bit. We're learning that from our customers, which is really nice. And it's always really nice to have it be successful as well. So some of our learnings are, what's the narrowness of the tip that can go in? So I can give you some examples here. We worked with folks who use something like Integra, right? And so the 300 microliter long, which is a much narrower tip, can go in there. So we have similar information like that across things like Beckman Coulter and all of the other sort of automation platforms that are out there. And so my answer to you is yes, because I think thank you to the automation folks who have thought about that problem. And so I think there's a choice of tips that are available to us where we know that it can be used to suck out most of the material, everything that we need, and it can fit into the narrowing that happens, but it's not your typical narrowing that folks are used to. We wanted to keep that a little bit open so that we could take out as much of the liquid because recovery is so important in these processes.
Yeah, for sure. Let's see, maybe a couple more here. So are the R50 96 strips reusable or single use only? Are there recycling options?
Yeah, they're meant to be single use only, Miles. So it is, the way I describe it is if there's a liquid in there and you've turned on the energy that flows through, naturally there's going to be ions that come. We do our best to make those ions as inert as possible such that the cells are not impacted by those ions. But if you have touched a liquid, the thought process, if a liquid has touched the well, the thought process there is that that well is now eaten up, right? I think it goes beyond just reusability. The contamination in this, to me, is too high. Having a couple cells left over or being able to try to clean that up, it's not going to work in our favor if we're trying to see small differences between our samples.
Gotcha. Yeah, no, that's perfect. So let's see. So for the high throughput screening workflow, what is the typical total kind of hands-on time from cell prep to readout for a 96 well plate?
Yeah, that's a great question. It's a little bit harder to do the cell prep and payload prep side of it. I think that's scientist to scientist.
Yeah.
Generally, though, your cells shouldn't be living, especially primary cells, shouldn't really be outside of the incubator for beyond sort of like, in my opinion, 30 minutes to an hour, right? So I would say that that's your prep of your cells. That's the max time they can be out, right? You should probably prep your payloads before that. Say that takes another 30 minutes. So you're probably looking at somewhere in the 1 hour mark for at least your payload and cell prep. Now loading is pretty easy. So if you're doing everything, in terms of prepping your cells, you got to spin it down, right? Like, so you've got to be able to remove the media and switch it with the electroporation buffer. So all of that should be done in that hour time frame, in my opinion. Your payload needs to be brought to the right concentration. That can be done outside of the cells. So if let's say you're putting that 1 hour mark for that prep, now everything's ready. It's already in the right size tubes. And theoretically, now it's just your job of disseminating it across the plate, right? So to me, that should probably take a total of 30 minutes, no more, right? If you're doing the full plate. And then running the actual samples, like we said, if you already prepared your experimental workflow before at your computer, take that time out, right? Because now you're just focused on preparing everything and running the experiment. So it takes you a minute to turn the machine on, click the protocol, because you've already set up your experimental plan. So click that experimental plan and hit start. It charges and discharges pretty quickly. Like we said, it's a 3-minute run, and now you're ready to go. Your samples are ready. So if I had to sort of guess, I think you're under 2 hours, right? Like if you're going from start to finish for a full.
Yeah, no, that's great. For sure,
Mhm.
yeah. Let's see, maybe this last one here. So what level of inter-laboratory variability is typically seen with efficiency and cell viability?
So, we can look at some of the slides if that makes sense. I would go with that reproducibility slide, if I really could there.
Yeah, you should be able to click it on the side there and bring it. There we go.
Yeah. So I think this is the variability that we see. So this is actually in T cells, again, primary cells, right? So you would see something even tighter, like we showed you with the K562s, which I think is not relevant, but I'll show you in a minute. So what you're seeing here, it's a single donor, but really we're just repeating this again and again. And what you're seeing is really that where the length of the dots are is that variability, right? So if you're looking at viability, you're somewhere in sort of like 10%, I would say, in your worst case scenario. If you're looking at transfection efficiency, again, you're in that sort of 8%, it looks like, in a worst case scenario. I don't know if my cursor shows, but this is the bar I'm referring to. So really, it's super tight. This is also across multiple users in our lab. So it's not just one person generating this data over and over again. So it's getting that user. So we held the donor variability, but it allows you to then propagate the ability, right? And what you're seeing is that really tightness there, which I think is really cool. If you go back to that K562 for anyone interested in immortalized lines, like you can see how tight that is, right? So those are your error bars, and they're super tight.
All right, awesome. Well, Grace, I think this is a great place to wrap up. And so really appreciate you being here today. This was, again, awesome to see. And thanks for getting all these questions. And audience members, thanks so much for joining us. If you did want to reach out to Grace with a question, you can still either enter it here, or if you go to her picture on the side of the screen, should be a little blue circle next to it with like a mail icon. Click on that. It'll open your email automatically, so you don't have to type in your email, which is also there on the screen. But click on that, it'll open an email, and you'll have that ready to go even after we wrap up the event here now. So thanks so much again to everyone for joining us. Appreciate you all being here today. We hope that you have a great rest of your day, and we'll see you next time. Bye, everybody.
Thank you.